Bueno, buenas tardes a todos de nuevo.
Good
Estamos ya
afternoon once again. This is the last session of the day, the keynote lecture that is going to close from a scientific standpoint today's session. We have lived a wonderful journey. We have really gone through every single aspect about immunotherapy. We have reached the final moment where we have discussed very different situations. And the best happy ending to this wonderful, interesting, fruitful session is the masterclass by Dr. Carlos Cordon. And we are extremely thankful once again for his time, for his presence today with all of us. You all know Carlos Cordon. He studied medicine at the University of Barcelona, and then he obtained a PhD in cell biology in Cornell. And then until 2006, he was a fellow member of the pneumopathology department, where he created the program of molecular pathology, and he became a professor at Cornell. And then after being in Columbia, he is a professor, a vice chair of pathology,
chair del Departamento de Patología
of the Department of Pathology and Self-Based Medicine in Mount Sinai Health System in the Lightcomb School of Medicine. I'm sure you'll know that it is impossible to follow the career of Carlos Cornell. However, we have probably seen him in several occasions, because in many occasions he has touched upon several topics. He has probably been one of the first people talking about the need to study the taxonomy, the specific molecular taxonomy of the different tumors, with a vast knowledge of tumors, analyzing potential biomarkers with new predictive cycles, new therapeutic targets. He has one of the first people talking, or at least me hearing him talking about system pathology. And lately he has been talking about the study of cancer stem cells, and during the pandemic he has even had time to develop some tools for the diagnosis of these. His scientific career has been very fruitful. He's one of the researchers in the world that has had more scientific research. He has more than 100 codes, ICH of 157. And without further ado, I'm going to hand over the floor to Carlos Cordon, who will most certainly tell you very, very interesting things. Thank you. Thank you, Luis and Carlos, for this kind invitation. It is a pleasure to be with all of you today. I'm going to share the screen with you today in order to see if we can be together today. The original title of the conference were Future Prospects of Immunotherapy in Cancer. But I would like to start by giving this overview about how relevant the moment is now, and the parallel between cancer and COVID-19, in a way that we perhaps want to share with you what we have learned. We still have a long way to go, but we have learned sufficiently as to handle these patients better with cancer and with COVID-19. What is the similarities that we are going to draw? Not only based on scientific data, based on clinical observations and other labs of basic research and clinical application. The topic I would like to develop goes as follows. I will try and summarize it in 3 or 4 very basic chapters. I would like to start talking about when the concept of immunomodulation started, when the concept started, and the functionality of being able to escape from the immune surveillance. What are the implications in cancer, and how have we learned to you know, get this type of machines in absence of mutations, and how the things that we have learned from the COVID-19 pandemic, both for the lab studies of pathophysiology, have allowed us to analyze the disease based on knowledge in the oncology world. And we will then finish by explaining what are the new improvements in the concept of COVID-19, the potential oncology implications, which are more profound than what we have realized so far. And let me just remind you that immunomodulation, real immunomodulation, is done through the HLA complexes, which at the end of the day is the machine that we have to present antigens, and all the rest revolves around the HLA, PDL, PD-L1. So basically, how this machine works, it is very interesting. The onset of any non-recognized antigen recognized as such, the proteasoma, as we explained afterwards, which is like a magnificent titrator that destroys in tiny little parts everything it sees, goes through several transportations in a specific space where the epitopes and the antigens that can be presented for our immune response are uploaded in the HLA molecules, which also start a whole projection from cytoplasma to membrane when they expose these epitopes. So the immune system recognizes them through different receptors where we can have an adequate immune response. The absence of a functional HLA, when they are bound together, HLA receptors and a natural killer, we have like a phantom cell. And when does this phantom start in our real physiology, where life starts, really? In 1988, it was proven that the embryo cell is a stem cell. All cells of all of us now come from our mothers. In forensic, we use the mitochondrial NDA. It's a direct, accurate line, one of the best fingerprints that we do have. And this cell, this is going to affect sperm, and this is going to create a new DNA load, needs the reduction of presence of HLA. Otherwise, the father's antigens will be recognized in the mother's endometrium when it be rejected. So none of us would have been born because we would be rejected as the recognition of parent antigens are recognized. In this study, I had the opportunity to work with Michael Hellman, who came to the United States for a sabbatical period. He was the chair back then of the Department of Immunogenetic in the Weizmann Institute, in order to gain greater access to several tissues. And where we started this collaboration in 1990, with the first publication published in 1991, when he asked me to study if the neoplastic cells, the metastatic ones, were formed with HLA or were they losing it. And this is the picture in 1991, one of the first pictures where we observed that groups of neoplastic cells, either within within metastasis, had lost HLA. And basically, this phenotype was associated with a tumoral progression and with biological and clinical aggressiveness of these cases. And on the flip side, well, many years have gone by and many years will go by. We have had the onset of new technologies and access through different laws to cellular and genetic material of human embryos. And in a study that we're about to publish, we're going to share what we had observed in the 1980s and 90s is now reproduced with modern technologies. And at the beginning of our embryo development, the groups that are less expressed are those who are really related with the entire immune system. So the HLA with modern technology, with profound networks, and at a certain moment having this regulation and these cells not being rejected. Since we have part of this in the embryo world with modern technology, we can draw a parallel similarity in these human clinical tumor cells. On all the publications of our group and other groups have proven that in all tumors, solid and hematologic tumors, the percentage of neoplastic cells which are in the tumor have the features of these normal embryo stem cell, which can be drawn the similarity. They do not have differentiation antigens. This is an epithelial prostate cancer with no cytokeratins. This cell does not produce other antigens such as PSA, and it does not have HLA. Therefore, they do have the same features as another stem cell may have. And moreover, they have a more asymmetrical division. In a culture, they have differentiated cells, keratin positive, androgen receptor that can release PSA. This is the work we have undergone for several decades that many have proven with new technologies. The negative phenotype has a lot to do with mutations or selective deletions in chromosome 6 or specific mutations, which allow us to classify HLA as a suppressor gene. This is the work approved by several groups, a collaborator, another friend, Dr. Toy Ribas, who through the passing of years has proven that HLA is one of the most affected genes in colorectal cancers, and certain specific mutations in such area where the antigen, the epitope is presented, and it makes it completely inert. In a most recent article, he has classified the different types based on the percentage of mutations and mutations of HLA and other genes involved in the antigen presentation.
And very recently, in one of the journals in Nature, evidence was presented immunohistochemistry level that they can present a partial loss of HLA or a complete loss. And this complete loss is more robust in metastatic cancer than in primary metastatic tumors than in primary tumor, and this is associated to a more negative phenotype of PDL, PD-L1. Therefore, we're starting to study, to recommend that perhaps for some immunotherapy studies, we should consider wider panels, perhaps other biomarkers such as HLA. And studies from our lab and others that have studied primarily metastatic tumors measured through time, because we've had material from biopsies or autopsies. We've seen that primary tumors, both with chemotherapy or immunotherapy, are highly populated with cells with these characteristics. In more modern studies, a study that's been prepared for published, we studied single cell analysis, and we realized that in sarcomas, in lymphomas, and carcinomas, there's differentiated cells, and there's compartment of cells that are HLA negative that have these characteristics and that are very similar to the ones we've studied so far. And one of the characteristics is that they have the TMDs lower than differentiated cells. They don't divide as frequently. So we continue to think that one of the important mechanisms to escape immune surveillance is efficacy of a system that if presents antigens or neoantigens to the immune system, the system will act. But if this neoplastic cell don't have the machines 100% effective, they can become ghost cells, they can escape an immune attack. At some point in this presentation, we're going to study in parallel oncovirus and SARS-CoV-2. This trend or tendency virus have to inhibit specific proteins and genes in cancers such as P53RB1 and P10 that are infiltrated in all the tumors that have machinery that can grind them or shred them. They have enzymes that can degrade them. Viruses have learned the same thing, and in papillomavirus, there are early proteins E6, E7 that inhibit TP53 and RB1, and there's an E6 and E7 because there's an E5, and E5 is the one that ubiquitinizes and destroys HLA. We're going to stop here. We're going to take a break and speak about what's going on in our world today, about this new virus that has arrived, whether it has characteristics similar to oncoviruses, what's the connection of this virus with viruses that have affected cancer patients, and what's the pathophysiology of this new disease. This is the enemy. Nature has created an enemy, a non-segmented virus, RNA positive, which is relatively complex, relatively big, produces 29 proteins, 4 highly structured, S protein, envelope protein, nucleus proteins, but there are 24 proteins that are critical. We're going to speak about them because NSP1 is non-structural protein 1, together with proteins 12 and 15, inhibit interferon, and the NSP8 inhibits HLA. So they have similar connotation to some of the oncoviruses. We've used this system, this model, to develop new tests. This is the enemy. Nature has provided us with antibodies that we can create ourselves and that heal at a higher level than when they can neutralize. They're targeted at this protein you see, the one that protrudes from the envelope, which is S protein, and more particularly the region in which the binding site is, which is the receptor ACE2. At different points in time in my life, I've had the opportunity to work with great people. We're going to present the work made by many collaborators, many hours of work. The microbiology department in Mount Sinai is one of the best in the countries. Here there are rankings of all the departments based on publications, impact factors, the number of grants, the number of patents, innovation, and the Department of Microbiology, led by Dr. Peter Paisi, a member of the National Academy, is mainly focused in viral studies and influenza virus. They have recruited wonderful people. One of them is Spanish guy, Adolfo Garcia Sastre, who has joined the National Academy of Sciences last year because of his works in virology and influenza. So when problems in China started to appear, some of our colleagues went there and we had access to viral sequences and to the production of antigens, perhaps before many other groups. And we started to ask ourselves what will happen. And when we saw that it was moving from China to other places in Asia and later on to Europe, we realized that the question was not whether it was going to happen or not, but when. So we prepared ourselves to take a deep dive into COVID-19 world, both from a diagnosis and therapy orientation world. When you're a pathologist, we expect pathologists to help us to have the appropriate diagnosis, an effective one, developing new lab tests that are highly specific and sensitive. That's what we did. We started to prepare primers for PCR, but we decided yes or no, which is what we decided that yes or no is not enough for us. This is what everyone is doing. It's not what yes or no. This is not what pathologists do. If you send me a blood sample, you don't want me to say that yes, it has lymphocytes, or you want to know if they have responding white blood cells. So yes or no is not enough. In other viral diseases, we've generated quantitative studies called viral load studies based on a number of copies per milliliter in the liquid of this where this potential virus is located when it comes to us. So it's not just a qualitative studies. We're going to do quantitative studies of viral load, and this has been very important from a clinical point of view. And we developed the same strategy to monitor our patients and the community. The pathologist helps diagnosis, monitoring of patients throughout many studies, and when anything goes wrong, we help to understand the disease based on autopsy materials. In the context of monitorization, we decided to carry out ELISA blood test, EC, and quickly to do, not expensive. They were qualitative but quantitative too in the number of antibodies and nanograms per milliliter of blood. This was very important. And then we decided to expose ourselves to be in the front line and to help understand this disease and the extent of the cell and organ damage that this virus can cause, showing that this is not a simple disease as we thought at the beginning. It's highly complex and a disease which can be compared to cancer. Let's start with the diagnosis. We'll continue with monitorization, and then we'll study the damage and the comparison with cancer. Diagnosis tests are PCR tests. They're obtained through nasopharyngeal swabs. When they arrive to the lab, they may contain virus, then we need to inactivate them with heat, 50 60 Celsius, and then RNA extraction or direct techniques for amplification. And most of these PCR studies generate amplification curves that start at cycle 18, 20, 22, and that finish at 34, 38. So this is the normal pattern of people who has the virus and it's detected through PCR. The negative cases in these red lines here were not positive. The surprise was when, right from the beginning, we started to see that there were people that started to amplify at the 18th cycle, and some individuals did not amplify until cycle number 36, 38. Some of my residents, my students thought that they were false positive and didn't understand. It was easy to understand. These individuals had a very high viral load. They started to be detected at the very beginning. These individuals had a very low viral load. And when we established the correlation between very sick patients that were admitted to our system, we realized that viral load was critical to identify patients that will have a very aggressive disease and they were going to die from it. What was our mistake at the beginning of the treatment of COVID patients with or without malignancies? We expected this disease to be similar to the other two diseases that preceded SARS-2, which was SARS-1, respiratory distress, the first one we saw 10, 12 years ago, and then the MERS disease. In these two diseases, more than 90% of the patients, when they were infected, had a temperature, had fever. So in Airburst, we established these little doors, temperature was measured, and we could identify patients who were sick with these viruses with a relatively simple test. And we stopped part of the pandemic because we had something that identified individuals immediately. A very significant number of patients infected with SARS-CoV-2 have no temperature. So when they arrived to our institutions, at least ours in New York, if they had no fever and they appeared to be okay, they were sent back home with Tylenol. 2 or 3 days later, they came back with the whole family because if the viral load was high, they're big spreaders. They infected relatives, infected them, and their disease evolved in a very different way than we expected. So the viral load has turned to be one of the most interesting biomarkers to use remdesivir and some antivirals at the beginning, not just in the middle or in the end when managing a patient with COVID-19. Now we have the diagnosis. What do we do with monitoring? Monitoring is carried out through serology testing. We did the same thing. We learned that if you start to look for antibodies after 14, 15 days, most patients are going to be still negative. The IgG response arises afterwards, after the third, fourth week. The paper published this week in Science shows that these levels are kept and can be effective in some patients. we used this serology testing, first of all, to know who had the disease, but at the same time, to identify individuals that have blood highly rich in anti-SARS-2 antibodies to take their serum and use it as a therapy measure. It's a very easy test to take the blood, you dilute it, and it's put in some plaques, and at the bottom you have the antigen, either the total protein or the binding site. And if there's an antibody that identifies this antigen, there's going to be an antigen antibody and a reaction that's going to generate color that can be quantified. Negative individuals have white color. Individuals with a low number of antibodies, the color is more or less yellow, and the higher the concentration, the darker the yellow color is. So optical density can be translated to arbitrary units and then nanograms of immunoglobulin per milliliter. This is what we did. Our test, which works with the spike protein S, has two parts. First one, the binding domain, and second with the whole protein. And there's a positive and negative value that are highly predictive. So it's a solid test. And in the study we've published this week, I think there have been several communications in different media in Spain today about this. We've answered three important questions. First one, everyone generates, but does everyone generate the same type of antibodies from the same number? No. Some individuals have low titer and other people have high titer. And titles are really important, as we're going to see in a while. Question number two, are these titles maintained through time or do they disappear? In our study, we can see that in most cases in which the titles are high, there's a decrease, slightly small, but in some cases, particularly individuals with very low titer antibodies can even disappear with time. And third question, first of probably the most important ones, are these antibodies functional? Are they protecting us? Can they protect us, all of us, in the same way? No, because we have shown through microneutralization techniques that low titers of antibodies, a low concentration of antibodies, produces only 30-40% of neutralization, viral attack killing the infected cells. So you need a concentration relatively high of antibodies, which are generated in many cases to be functional and to be able to say that our community is immunized or that has immunity. This was important because when selecting individuals to get plasma to be provided to other individuals who are highly sick, if we selected individuals with very low levels, this plasma was not useful for patients, and perhaps some studies are not working because they did not carry out quantitative statements and they used donors that had very low levels. In our case, we didn't accept them. We only accepted individuals with high levels of antibodies. And when we applied this convalescent plasma to highly sick individuals, we had just a sample of 10%, but patients without this treatment, their mortality was higher. What did we learn from monitoring? That similarly to diagnosis, yes or no can be a good diagnosis for a rapid test, but if you need monitoring and stratification studies, we need quantification both for the viral load and the content of antibodies. What did we learn? Learned this disease is not the typical disease I was expecting when I was with my microscope in the optoscope room. I'm going to show you data that show the heterogeneity of this disease. This is the typical case of a viral respiratory disease. A woman, 56 years old, no other comorbidity, low temperature, 37.3. Perhaps this is not a temperature, respiratory problems, but with inflammation indicators skyrocketing. CRP is 270, ferritin, which has to be below 100 to 100, it's at 700, etc. Temperature the patient starts to fare badly. Part of the complication was a respiratory one, we'll see in a while. Patient reaches oxygen levels that are incompatible with life and ends up dying from the disease. And in the autopsy, we realized that her lungs were not highly congested with inflammatory cells. And really, this patient had more lymphopenia than lymphocytosis. We realized that the heart has fibers that start to show heart dysfunction. And we asked ourselves, where did these cytokines and this inflammation come from? And we realized that one of the most prominent cells in the bone marrow, the lung, etc., well, macrophages activated and phagocyting. It was the macrophage activated symptom, which is common to other diseases such as Kawasaki, the classic Kawasaki. This is a lung from one of our COVID patients who also had a bacterial pneumonia, so that you can compare how the spaces look here compared to the same spaces of the viral pneumonia in COVID-19. Here you can see the spaces. Here you don't see the spaces, they're saturated. This acute and chronic inflammatory infiltrate. And here you see the hyaline membranes that prevent the oxygen flow from the alveolar space to the blood vessel. It's a vessel proliferation similar to the angiogenesis that happens in some tumors. This patient end up dying with a pulmonary failure, which in turn causes multi-organ failure due to the lack of oxygen. Through electron microscopy, we proved that cells and epithelial cells, here they're enlarged, and at the same time, this was beyond what we were seeing at the beginning. They were not just in epithelial cells, they were in some blood vessels. There were problems, and we saw some endothelial cells with viruses and infected. The macrophage system was highly activated. It's what we call an activated macrophage syndrome. We assume that in some patients, the virus arrives, enters the respiratory system, and then the epithelium, which is enlarged through the cytokine release produced by the macrophages. But other patients had different symptoms that were respiratory, but it was rather neuropsychiatric and cardiovascular. Some patients said, I have memory gaps, I'm an English teacher, and I don't know how to write some words. I don't remember what I did this morning. And we started to realize that there were some patients that had delirium even. This is a patient, 68 years. They arrived with a temperature of 37.1 degrees. I don't think this is a temperature. Temperature is not a good indicator of this disease. I've shown cases that when they arrived, they were very sick, but they were very sick, but they didn't have a temperature. They ended up dying. But inflammatory markers are skyrocketing. The patient has distinct different complications, and he ended up dying. In the autopsy, something caught our attention. We took a look at the brain, and there were many clots, microhemorrhages, and thromboembolisms. This is a section of the right hemisphere of the brain. As you can see, there are multiple areas of micro bleeding, embolisms that were present in mid-size and small size, but also big size vessels in the brain, in the surface of the brain, but also in the parenchyma. So we asked ourselves the following question. Perhaps the cells are similar to the ones we have studied in the past for cancer, part of the brain-blood barrier. Perhaps endothelial cells in the brain, when realized the specific phenomenon, we saw that the glycoprotein B was present physiologically in endothelial cells in blood-brain barrier, but not in big vessels such as the aorta. Perhaps some specific vessels in our body have specific receptors for ACE2, and realized that this was the reality. Endothelial cells in small and mid-sized cells were very positive for this. And this gave us the opportunity to study these brains. We didn't just see bleeding, we also saw clots that caused strokes. There were big areas of cell necrosis that macrophages were trying to solve. At the same time, since they were there, they were creating further endotheliitis, so they were not helping. And this thing that we saw in the brain was also seen in the heart, other organs such as the liver, but not other organs. And that's why we suggested that as a neoplastic disease, COVID-19 virus entered through the epithelium, created cell apoptosis, entered the blood vessels, creating what we call metastases. Although we only use metastases for tumors, when I explain pathology to my students, I always have to remind them that metastases come from Greek. It means going at a distance. And this phenomenon that happens mainly in oncology also happens in other diseases in which the pathogen is not a transformed cell, it's a bacteria or a virus. And this needs a physiological sense. In the case of cancer cells, they circulate, but they have a place to stay, which are matrix proteins in different organs when they can create colonies and develop. And in this context, SARS-CoV-2 virus has receptors, which are ACE2 receptors that allow them to bind and create the subsequent pathology. And this gave rise to some studies by Dr. Valentin Fuster, and together with us, he saw that this phenomenon could be tackled, and we started to treat very sick patients with anti-clotting agents. We were afraid that this patient could have vessel-related issues or bleeding. Some cases were solved with prophylaxis therapy. We rescued more than half of the patients that with modern anti-clotting agents, they had a better result. Right now, Valentin has a clinical program in many countries to have solid data to adjust the dosage for these patients. So this week, another study has been published in Cancer Cell. The publication in November is going to be exclusively devoted to COVID and cancer. We suggested the publisher and other colleagues to have a stage system as we do with cancer. Why? Because stages allow us to put in a single roadmap to know where we're headed, not blindfolded. The pathophysiology, stage 1, stage 2, 3, expansion. The pathophysiology, together with the best test for diagnosis and monitoring, and therapeutic interventions. So what we started to say is it is not right for a diabetic or neoplastic patient to be sent home with an aspirin or Tylenol if the viral load is high and if the disease is advancing from a neuropsychiatric point of view, or there are clotting issues. This can be helpful for managing COVID-19 patients. And also for policy purposes, we can go to lobbies that can help us to understand what's going on. State system exists in other diseases, chronic renal disease, for example, and this gives some positivity to patients, or at least anxiety is reduced, particularly in patients where you can show that they're in stage 1 or 2 and their progression is not going to be as complicated as in stage 3 or 4. So, to finish my presentation, I would like to say that in the context of COVID-19, we've realized at the beginning it was conceptualized as a respiratory disease, but it's much more complex with endotheliitis, with coagulation issues, with macrophage activation syndrome that produces a prominent immunological response that further accelerates vasculitis, and coagulopathy and hypoxia become severe. Multiple organs, and in many cases, the patient dies. There were very important barriers for cancer patients during this pandemic, and they still exist. Barriers related to access to studies in which they were, access to treatments, to therapies. A sudden stop in the healthcare system to continue with the clinical trials. Patients with cancers, many of them have a high risk because they have comorbidities, they have cancer. And some of them, their immune system is severely affected. Patients with cancer, we've seen in some studies, and more studies are going to be published in November in Cancer Cell, their progression is faster for COVID-19 and their own disease. And patients with lung cancer and hematological malignancies have a more adverse progression and clinical course. It has been proven that the receptors for SARS-CoV-2 in the lung are increased in smoking patients and patients with lung cancer. We've realized that there are different approaches that can be helpful for our patients. We've carried out different studies to combine the study of cancer and COVID-19. One of them is the fact that the machine SARS-CoV-2 needs to enter the cells. It's a machine that some of you and us pathologists are familiar with, and is as follows. The signaling system of androgens makes this machine to bind transactivity through effects genes, but some genes, one of the genes is TARMS2, which was described by Aaron. It's one of the genes that's translocated in prostate cancer. There are many studies regarding TARMS2 inhibitors in clinical trials with prostate cancer. How does it work? This protein, which is an enzyme, goes to the membrane, and when it arrives there, it helps to produce this cut that you need to separate the part in the membrane of the virus to enter the cell. So AC2 needs to be cut through this serine protease, which makes the flexible part to enter the cell and stop the infection. We know that the androgen receptor is highly regulated at hormone level and age level. We've studied the nasal epithelium, the bronchial epithelium, and the lung epithelium of different ages and genders, and we realized that children to 7-8 years, they have virtually no AC receptors or TAMs in their lungs. So that could explain why they're not, the infection is not that high or the clinical course is different. And the androgen receptors is, you start to see it particularly in males after 14, 15, apparently it stabilizes at 20. And in young women, there's a huge difference in the presence of ACE and TRAMS2 in the lung. And we've seen this in different organs. Now we are studying with Arun Shanyanya the repercussions of this, and we're using autopsy materials from different diseases to establish a link between the androgen signaling system and what's happened with the AC receptors and the androgen signaling pathway. Like some viruses that generate proteins and inhibit the immune system, our group and others have proven that there are some proteins in SARS-CoV-2, non-structural proteins such as NSP1, 3, and 15 interfere with IFN induction, and NSP8 that downregulates HLA, and inhibits HLA, and also NSP3 helps the machine degrade P53, and NSP7A does the same thing. This is a diagram from one of the publications from our colleagues. This is another paper that has been just published where it's shown that non-structural protein 8 is highly linked to a reduction of HLA levels, and these HLA levels are neutralized and disappeared. I said before that oncoproteins of HPP 6, 7, and 5 are oriented to TP53, RB1, and HLA, and SP3, 7A, and in this case it would be 8, my slide is wrong, would decrease the levels of HLA, RB1, and TP53. What are we learning? We're using the same cancer principles of the transcriptome map that has been internationally produced for cancer to study COVID-19 in both blood vessel development and activated macrophage system and cytokine production and other things are very, very similar in chronic patients. And at anatomic levels, the location of the proteins are going to be very similar in this context. So this shows that stages are critical in complex diseases, and as in cancer, we can use stages, a stage system in which the pathology, lab tests, and therapies can provide us with the view on how to manage these individuals. If we combine this with stage in stages in cancer, it also be helpful to manage cancer patients with COVID-19. And I'm going to stop here because I think we've given you a lot of information. I hope you can digest it. If there are questions, I'll be happy to answer them. There's a lot to be learned. I think we've learned a lot. I don't think we're applying. I think we are applying what we've learned so far in a more disciplined way. Thank you, Carlos. I think your conference was wonderful. Seeing this comparison between not just the diseases, but the mechanisms in which the disease is produced, I think this is surprising. And it has caught my attention. If we can ask a couple of questions regarding this. Yes, yes. We need to take advantage of this opportunity. One of the problems you have mentioned is that the clinical course was worse. In our case, we've seen mortality around 35%. In hematologic tumors, it was over 40%. One of the things that we asked ourselves when treating patients that need chemotherapy is whether we should give them a ablation chemotherapy, or for example, lymphoma or an SCLC, whether we need to use GSCF. It prevents immunosuppression, but you're afraid that lung inflammation, if there's an infection, well, it could be worse because obviously you're enabling myeloid inflammation. What would you use?
Yo me quedé sorprendido cuando
I
empezamos
was surprised to see when we were carrying out autopsies. I expected to see a brutal myeloid response, lymphocytes everywhere. We were the only ones that we've seen this. I started to explain it because if non-structural proteins diminish the production of interferon, lymphocytes are going to be asleep. So what's left? Macrophages. We've seen macrophages that are very angry, they eat everything, they're everywhere. I think that part of the problem of this cytokine release, this brutal inflammation some patients have, is due to the macrophages rather than the neutrophils. So I think that applying growth factor to stimulate these colonies would probably be good for the patient. As a matter of fact, I haven't seen that combination is inflammation and neutropenia. That's why we do it. That's reality. And as you've seen, in some cases of patients that die in our hands, this phenotype, I think we haven't been sufficiently aggressive, perhaps because we didn't know. We didn't have these opportunities with antibodies and anti-inflammatory drugs, cross-stimulating factors. But now that we know that this is good, we have the evidence, I think we need to use it before rather than later. In oncology, we've learned that chemotherapy and immunotherapy are not just for people in a really advanced disease. When we apply at the beginning, we're really helpful. I think it's the same thing for COVID-19, and particularly in cancer patients with COVID-19, because they're immunosuppressed. In the case of lung cancer, because they have AC receptors more expressed than in a normal case due to the age. And I think it's a good therapy orientation, doing this at an earlier stage. Last question is, when you've explained the comparison, perhaps in the COVID infection, P53 and other proteins disappear through a different mechanism, not a mutation. You've explained HLA right from the beginning. Are you familiar with a paper by Charlie Swanton Group that shows that one of the first thing that disappears is HLA, the inflammation of the lung? The second thing that appears is that clonal mutations, the one that create new antigens, with time, these new antigens, there's a downregulation, which makes sense because the main proteins always persist. They're important to not to call the immune system. Have you seen if any of the antigens, the important antigens of this virus end up downregulating this, so there's no This is based on the single cell analysis. And yes, we are seeing this. We see that infected cells have an absence of HLA, they start to have problems, and we're worried because we can speculate since we're among friends. But imagine that loan holders, well, we don't know what's going to happen with them. There's like a brain dysfunction for months. Their abilities are diminishing. I hope they don't, but we have to be very vigilant, particularly with children and young people that have many years ahead of them. Let's hope that this virus does not transfer. It has to be like this. We need to pay close attention to some populations, particularly young people with comorbidities, diabetes, autoimmune diseases, because if any cell is left behind, damaged with these essential proteins, they can give us nasty surprises. Dr. Pazares, could you please turn on the light in your room, please? Please, just a comment, a question. I don't want to miss this opportunity. This thing you've just said is frightening. The secondary effects, the side effects that we are already seeing in patients, unexpected effects. But you've opened a door to a very worrying world. I know that there's no answer. I wish we had it, but why some patients in such a dramatic way and others not so much? You've mentioned the receptors, children, smoking. There are many parts that are not explained. The danger, Carlos, I don't know if in the US it's like this, but maybe in Spain, we are carrying out tests that are not providing us sufficient information regarding the infection status of our population, because perhaps we're identifying parts of the virus, but not virus really active. This generates confusion, alarm within the dramatic situation that we have with this disease. It is true if there are no lab tests that have a high specificity or sensitivity, saying yes or no is not going to be helpful because the level of this is so negative that we could even disappear. In the case of viral studies, the fast antigen tests, I think they're complicated. false negative is very high. So for viral molecular diagnosis is horrible because they test negative and they go to the grandparents' place, they sneeze, and everyone's sick next day. So they should be minimized. We don't know if the viral load is high or not, but we don't know how to manage this patient. In the case of monetarization, the bad thing is a false positive because if you have a test which is not quantitative and you say, yes, you're positive, and the truth is that it's not positive or it's positive at a very low level, the person thinks that it's immunized, helps the patient, etc. And the next day, he's sick and his whole family is sick. So tests that are being offered don't have the specific sensitivity that we think it's enough. None of them is quantitative. Everything that's qualitative leaves it open to interpretation. Hippocrates said that science was developed in two ways, either opinion or experience, and opinion was based in ignorance. We don't want to insult anyone here. If we have better tests, why don't we use them? And if we can study the patient with tests that are home, they're not complicated, why not?
Economía 101 en Estados Unidos, el primer curso de
what we call Pharmacoeconomia 101 in the US, they say that the management of a chronic patient test, when you study the cost of the patient, tests are just 1%. The cost is basically the treatment, the anxiety, and all these things. Investing in good tests right from the beginning, such as interleukin tests, anti-inflammatory tests, they would add much more to the management and they would decrease the number of serious cases. I would like to give this sentence. If I was a journalist, I would use it. An investment in diagnostic tests is what will allow us to maintain the universal health system in Spain. This means regarding molecular diagnosis in Spain in all its aspects. And it's not understandable. Okay, Luis, please, could you close this session? And I think that we'll need to close. No, my last words are to thank you. I'm sorry. Someone's calling me. I hope it's not the dean. Thank you for this opportunity to share this moment and this work with a huge team of colleagues, including Spanish colleagues such as Valentín Fuster and Adolfo García Sastre. Thank you very much. It has been an inspiring talk. Data are very important, but the view, the explanation of the mechanisms why things happen is really inspiring for many of us, and I'm sure the young people will love that. I just wanted to say that I'm going to send you a PDF with the presentation so that you can share it with the colleagues that want to see these things in more detail. Thank you very much, Carlos.
Su participación en esta sesión y te dejo ya para finalizar.
So we're going to close the session and then you close the agenda. We're going to close a meeting that has lasted almost 11 hours or 10 hours. And this is complicated. Complicated because people are already exhausted. I hope there are many people still with us because this last presentation was wonderful. Eco Foundation fulfills its role with this type of sessions. What's our mission? Providing Spanish oncology with sufficient tools to grow in quality, in improving care and research, and why not, in this difficult time, education in our specialty. Who would have thought when ECHO Foundation was created 11 years ago that we would speak about immunotherapy for a whole day in several panels about the increase of survival and why not the curing of lung cancer patients with metastases. 5 years ago, 10 years ago, this was unthinkable. I have to thank the speakers for the presentations from 5 different countries: Italy, Israel, the US, etc., Portugal, for your participation in a globalized world in which knowledge knows no borders, in which there are many challenges to move forward to cure cancer. And little by little, we sort problems out: immunology, immunotherapy, and specific molecular actions. I would like to congratulate the national and international collaboration groups, particularly the national ones, that have allowed scientific breakthroughs in our country. I would like to say something which has to do with what's going on with cancer in Spain. We're in the second wave now. In the first wave, we had to adapt to something that was unknown. And in the second wave, I think that we need to take note of many things that we've learned, and we need to be really careful with our cancer patients. And we need to try to apply our current knowledge on the repercussion of this infection in the survival of cancer patients and use new strategies to face them. Our patient survival is at stake, and I don't want to be too dramatic, but I would say even the survival of our own specialty. Dr. Carlos Cardon has said it. During the pandemic, in the US, the number of patients that were included in clinical trials has been reduced. There's a recent publication that said that there was a decrease of 40% in the inclusion of patients in clinical trials. I think that altogether we're strong. We have a lot of knowledge and ethics to take care of our patients. We have many colleagues that are great professionals and are working in the US and other countries that can contribute with knowledge. We need to count on them. This afternoon was a stimulating conference from an intellectual point of view, and on behalf of the foundation, I would like to thank the speakers and the participants for this ability to assimilate so much knowledge. And then an optimistic message, because we are reaching high survival levels in our patients. Immunotherapy is helping us to save lives. Immunotherapy is a tool that has reached us not as a surprise, but the results are surprising. In many tumors, it's a very universal treatment. And well, it's already very late, almost 7. We started at 9:30. And I would like to finish by thanking all of you, and particularly the pharma companies that allow us to hold this type of meetings that are for training purposes. They're constructive, they help us position ourselves and consolidate as physicians to treat our patients. Without this help from the pharma industry, I don't know where we would be in oncology in Spain or medicine in Spain and in other countries. So I am deeply thankful to the Spanish pharma industry. So without further ado, I'd like to say goodbye. Good evening to all of you. I think we'll see each other soon because with so many trials, there are many meetings necessary. And this online setting has not reduced our possibilities to interact. We have increased, we are reaching to more people and farther than before. So goodbye, a warm goodbye.